figure this. Do this. Okay, there we go.
Do you go live very often?
Not really, no.
I didn't think you did.
Let's see. My live isn't working. Okay, there we go. I think that's good. All right, cool. Here I am. Here is Jonathan. Did you go by Jonathan or JJ?
Jay is fine. Yeah, Jay is fine.
Jay? Okay, cool. All right, so I'm here basically because I made a video about Geert Vanden Bossche, and you responded to it on your live stream. And you wanted me to come onto your show so that we could have a chat, I guess. So, do you want to introduce yourself or say anything about that?
I just wanted to say that I think we're two biologists trying to figure this out, and I did see you have seen your YouTube channel a few times, and one of my viewers brought to it the attention that you had done another critique of Geert. And so I watched it, and there were a couple things that I think you got wrong, and so that's why I did the live stream about it. And some of my viewers then actually were the ones who emailed you and encouraged you to come on, and so I'm happy you did.
Mm-hmm.
Is there a particular place that you wanted to start? Did you watch the live stream? I mean, do you have particular points that you have contention with, or
yeah? So I mean,
the whole thing was wrong, or
yeah. So I watched the live stream. I guess we can run down any points if you don't have a particular preference for starting.
No.
What's that?
No, go ahead, please. I would encourage you to do that, because I think if you have a particular set of sticking points, it would be easier, because I mean, I said what I needed to say in the live stream. I think the only thing I got wrong was that I said that Geert was referring to natural antibodies as being the complement system, but it turns out he was talking about IgM.
Right, so we can start there. So
Okay.
IgM, right? So that's probably the one part of the video I wish was clearer, and I think it was kind of me not understanding what Geert was talking about. I'm just gonna call him Geert sometimes because that's what my brain does. I know it's Geert.
Don't worry about it. That's fine.
But yeah. So I guess I'll just start by saying I was wrong to say or give the impression that there's no such thing as natural antibodies. What I was trying to really get across, and what I think Geert really kind of got wrong, was this idea that these natural antibodies can neutralize and clear a SARS-CoV-2 infection all on their own. So I guess I can explain the reasoning behind that if you want.
Sure, go ahead. I know exactly where you're at, so I'll just go make your whole point, and then I'll try to counter or at least interpret it.
Sure. So thanks to how antibodies work in the germline, we have antibodies in our body right now that could recognize, theoretically, any epitope of any pathogen. That's due to hypersomatic mutation and shuffling of VDJ genes. But those antibodies are not from class switch mature antibodies, so they're typically really low in abundance, and they're not going to be very good at binding. So you can detect them in cell assays. So there was a really nice paper in Science lately that was talking about germline antibodies that recognize SARS-CoV-2. So they exist, they're a thing, but they, as I said, they're in low abundance and they're not good at binding. So to expect them to neutralize and clear a SARS-CoV-2 infection on their own is not really good advice, especially when he says it the way he says it, as implying that a natural immune system can just deal with SARS-CoV-2 no matter what. That's the wrong impression to give. The antibodies need to undergo, the B cells making the antibodies that might bind SARS-CoV-2 need to undergo class switching and further hypersomatic mutation in order to create better antibodies that can actually, do something more against SARS-CoV-2. IgM is typically not going to do much against a virus. So, yeah, a little bit of confusion on my...
In that point, I think the thing that you're missing there, or at least the point that Geert is trying to make, is that when T cells are responding early to infection, they aren't responding to spike protein epitopes. They're responding to the earliest expressed proteins in viral infection. And when they then activate apoptosis in those infected cells, there are viral particles and fragments of these digested cells and the viral particles that get digested by the macrophages that are there, that are then processed, and those need to be cleaned up. That's what the antibodies do in a natural, well done infection. The antibodies are cleaning up the viral debris. They are not the primary way that the body is stopping the virus from infecting cells. How would an antibody that's in your blood stop a virus from infecting the epithelial cells of your lungs? It won't. That's what T cells do. And when they fight the infection, they create debris which the body has to clean up. And that debris is oftentimes very much the same from infection to infection. And that's the reason why those IgM antibodies work at low concentration and low affinity. And the only reason why you need to enrich antibodies for specific epitopes is when the viral load reaches a level where it's systemic. And that doesn't happen with every infection because T cells take care of that. And I think most of the TV biology ignores the fact that asymptomatic infection in a healthy person is T cells responding to the first genes expressed in viral infection, and that's not the spike protein.
I mean, the spike protein is the first thing the body sees when the virus enters.
That's not true. The first thing that the body sees are the proteins that are presented on the HLA receptors when the cells are infected, and those proteins are viral proteins. That's how it works.
Including spike, but spike is...
No, it's the first proteins that are expressed. Spike comes afterward. You first have to express the proteins which hijack the cellular machinery of the epithelium, and those proteins must also be expressed on the outside, and they are the first T cell epitopes available. There's no other way to debate that. Those are the first and most prolific T cell epitopes. They are identified in at least 10 different papers as the most immunogenic epitopes in the genome, and that's because T cells respond to the earliest proteins.
Okay, so before we get on to that, just getting back to what Geert said. What you said is not really what he's saying. He's saying that antibodies can clear a SARS-CoV-2 infection, natural antibodies, IgM pretty much alone. That's not correct.
I think that you're parsing things out, and he's a 20-year immunologist, and he knows that T cells are the primary response. He's read all of Stanley Perlman's papers. He's not. I mean, there's 10 years of data from 4 different labs that made their careers on defining T cells as the primary response.
He doesn't seem to think T cells are going to do much.
He can't, because he's a vaccinologist. Don't you see? You're falling in the same trap that he's in.
What are you talking about?
Vaccinologists have to look at antibodies because it's the only readout they've got.
No, you can measure T cells.
You can, but you can't measure all of them because the memory T cells reside in the lungs. And anybody that works on the immune system of a mouse knows that. You have to sacrifice the mouse to get the memory T cells. You can't measure them in a human. You have to get them when they're migrating.
No, you can get them from germinal centers.
You can get them, but they are not the memory cells. They are not the ones that are residing in the lungs. You can't. It's just not possible. You can argue, but there are no papers except for in mice.
There are. I mean, there's just a paper that was pretty recent, published in, where is it? Science, I believe. Yeah. Where they did that. They looked at longitudinal memory against SARS-CoV-2 in both vaccinated people and natural immune people, and they measured memory responses of B and T cells. and they
right, and
found
they're stimulated memory B cell responses. That's not the same thing, and you have to
memory
understand that.
memory T and B cells.
They're not the same as the ones that are residing in the lungs. You just are unaware of the fact, maybe, that there are memory cells which migrate back. There are memory cells that stay from clonal expansion and from antigen affinity enrichment, and then there are also memory cells that reside at the site of infection. There are
germinal and in germinal centers, which you can definitely assay.
Right, but you can't assay the ones that matter the most, the ones that they only understand from mouse experiments and monkey experiments, because they can remove the lungs. They can sample from the lungs. You're arguing against nothing though, because you're still ignoring the fact that you're saying T cells, but that you're not admitting that they do the job. What other cells are getting rid of the infected cells?
Macrophages?
No. Yeah. Macrophages don't get rid of infected cells the way that CD8 positive cells do, because they cannot target. They clean up. Exactly. So the primary response to any viral infection is CD8 and CD4 positive cells. Would you at least admit that? It's not antibodies. It's not antibodies, dude.
Well, no, antibodies are a frontline defense. If the
they
titer
are
is
not
high
a
enough,
frontline defense, because they do not reach.
If the titer is high enough, yes, they are.
That's
they
exactly
neutralize.
Why the titer does not stay very high in a healthy person, because
if antibody titers are high, the virus will not be able to infect or spread very well.
Is that what you see in the data now? That antibody titers correlate with people who can get? That's ridiculous. You know that's not true.
We know that neutralizing antibody titers correlate with outcome and with infection and everything.
But you know that I've done like 6 months worth of streaming to show that that's not true. There are paper after paper, dude. You don't look at any papers. You don't show any papers in your streams.
What are you talking about?
Crazy.
I link papers in every single one of my videos. Look.
But you don't interpret them correctly. It's kind of frustrating. You are trying desperately to sell the idea that antibodies are the thing, but they're really not.
Yep, they're really important.
But they're not as important as you think they are. Would you like to hear a virologist admit that? Like,
what are you talking about? They're
like a virologist say that they don't know what antibodies mean?
What are you talking about? They don't know what antibodies mean?
They do not know whether antibodies are a correlate of protection or not. And that's because
there's a difference there. Okay. A correlate of protection has not been determined for SARS-CoV-2. So that means that if you have really high antibodies from, say, a natural immune infection, and also really high but lower titers of antibodies from a vaccine, then that difference we don't know the significance of it. We don't know the immune correlate of protection, but we do know that higher antibody titers do correlate with better outcomes for patients.
It's very fascinating to me how convinced you are of this. It's very fascinating.
Okay, so what was wrong about that?
It's just that I can't find papers that show that antibodies to the spike protein are correlated with better outcomes. I just can't. Not the way that vitamin D is, not the way that zinc is, not the way. I mean, it's quite comical that you even laugh about that. I mean the unhealthiness of America versus the antibodies of America. It's comical to me.
Okay, so the immune system needs to be trained in order to be best protected against these things. Antibodies
it's never seen a coronavirus before.
Not SARS-CoV-2.
Oh, I see. And so what's so different about SARS-CoV-2 from the rest of the coronaviruses? Is it all the proteins or just a few? Do you even know?
It's very different. The closest.
How different? It's 61% different. And what's that 61%? Is it all spread out evenly, or is it found in certain proteins that are highly conserved across all coronaviruses? Or don't you know that answer?
I know that it's a 60%.
Mostly
a 60%.
60%.
A 60% difference in the genome is huge.
Yeah, it's huge
It's
unless
huge.
unless it's not all spread equally, my friend. And it's not.
But
It's not.
it
It's
doesn't
spread.
matter.
It
It
does,
doesn't
because the
matter.
immune system remembers the genes that don't change, genius.
Yeah,
There's
but it
evolution
doesn't.
here.
It's clearly not able to recognize and clear SARS-CoV-2 very
So
well
how
when
come
millions of people
50%
have
of the
died.
people, how come 50% of the people who get infected don't even get sick, don't even know they're infected? How do you explain that? That
Because
doesn't happen with flu. That doesn't
Yes, it-
happen with,
yeah, it
does
does.
it? Do you get high viral loads of flu and not show symptoms? You do not. Sorry,
Yeah.
but you don't.
Yeah,
That's
in some
complete
cases you do.
baloney.
You do.
It's
It's very common to have an asymptomatic carrier of something. That happens.
It does happen. Yes, it does happen, but it's much more likely to happen now that we have these silly transfections going.
What?
Yeah, transfections. You know, the
Yeah, I know what a transfection is. It's a choice.
Making
It's a
people-
choice to call the mRNA vaccine a transfection. But
it's not. That's what it is. It's not a
it's-
vaccine. It's a transfection. Vaccines have an
No.
adjuvant and a pathogen in them. They have an adjuvant.
these
and an
mRNA
antigen.
vaccines have an adjuvant. It's the mRNA.
The adjuvant is an mRNA. They designed
Mhm.
it specifically not to be immunogenic. Did you
Oh,
not
no.
read about how they did it?
Oh, I read about how they did it, and it does act as an adjuvant because they're able to purify it with HPLC that increases recruitment of dendritic cells to the site. They also pseudouridylate it, which also increases dendritic recruitment to the site. That acts
Oh
as an
my
adjuvant.
gosh, you are actually totally wrong. You know that, right? It's actually the opposite.
You're just saying I'm wrong, but
No, but
That's what happens.
I've interviewed both Robert Malone and Luigi Warren. Luigi Warren is one of the guys who invented this. He's the
Okay.
guy who came up with the the idea of using this alternative. Wow, man, you are shocking me right now.
I mean, that's literally how it works. Pseudouridylation and HPLC purification increases dendritic cell recruitment. I don't know.
Of what? Where at? You mean at the site of injection?
Yes.
Because the mRNA acts as an adjuvant.
Yes.
I would love for you to give me a reference for that. I would love for you to give me a reference for that, because
Okay.
it's my understanding
Uh-huh.
that the reason why they needed to use this alternative base is so that it would not be immunogenic. Because RNA is one of the most immunogenic things in the body.
Yeah, they went through a lot of trial and error.
mRNA.
Yeah, they went through a
Foreign
lot.
mRNA.
Yeah, they went through a lot of trial and error to get this right.
To get it not to be immunogenic, and now you're telling me it's the adjuvant. It's completely wrong.
No, that's definitely how it works.
I've heard other people tell me it's the lipid nanoparticle, it's the adjuvant. You guys are crazy.
How is no.
There is
It's
no adjuvant. That's the whole point of this. If the
It
mRNA
is
was an adjuvant, you would have such a problem.
Okay, let me just get this reference for you.
Is it from Moderna or Pfizer, or are you giving me a scientific reference from
No, this is a review of mRNA vaccines before COVID existed.
Cool. Send it to me, please. Do they talk at all about the fact that they had to reduce the immunogenicity of the mRNA, or how do they?
Oh yeah,
deal
they
with
had
that
to.
question?
They had to try to figure out how to make it stable enough that the immune system wouldn't totally just chew it up right away. But
But the immune system doesn't chew up mRNA, dude. You're a molecular biologist. You know that that's not the immune system's job. There are just RNases that do that. That's
yeah,
not the immune system.
it's passive. It's passive immunity. I get it.
Okay. Yeah. Okay. Well, I mean, just don't say that it's like the immune system. That's silly.
It's a defense against foreign invaders. That is part of the immune system.
I'm so confused. I'm so confused as to how this could be the adjuvant and also not immunogenic. I don't get it.
It's a balancing act.
It's not.
The immunostimulatory properties of mRNA conversely can be increased by the inclusion of an adjuvant, blah, 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 blah.
An inclusion of an adjuvant. You just said the words, you dope.
Oh, I'm reading.
What are you reading?
I'm
Oh my
finding it.
God. You just read the words and you're telling me I'm wrong? Find another paper, dude.
Well, I mean,
Holy shit.
Chill out.
Dude, you're starting to bother me because nothing you're talking about is right. It's the polar opposite. It's like you're just watching television, regurgitating it to me.
Yeah, that's a choice, so to call everyone you don't like TV scientists.
I'm calling everybody who's wrong. Everybody who says the same thing that PBS NewsHour says is a TV scientist, so I guess you are. I really want to be nice, dude, but this can't be true. You cannot have it be...
You're not really trying very hard to be nice.
You cannot, but you can't have it be not an adjuvant and an adjuvant, or not immunogenic and immunogenic. It's like you just read the article today, and you don't understand it.
You can have it. It's a balancing act.
You just read
things.
the text that said mRNA can be added with an adjuvant, and then you stopped reading it because you knew you were putting your foot in your mouth. Are you kidding me?
I'm trying to look for the right paragraph as you yell at me.
Try to look.
So
you should know it already, because I'm talking about general principles of immunology, and that is that mRNA is immunogenic. And you're telling me that this is the adjuvant. That can't be true, because otherwise the vaccine would not work like it's supposed to, which is expressing a ton of spike protein so that the body will build antibodies to the spike protein. It's one or the other, dude. It's crazy. It's like you work for Moderna and you're tripping over yourself to give a stock portfolio presentation.
Sure. Yeah, so that's exactly what they do. HPLC and pseudouridylation recruits dendritic cells.
What? You just said the same thing you said before. You're not explaining
it's
to me
in
the biology.
it's in this paper.
What is the biology behind it, though? Explain that to me. How does that work? That the mRNA gets in disguised in the lipid nanoparticle, so then it doesn't attract the immune system and it expresses spike protein, and then it attracts the immune system? How? What's happening? Can you explain that? Because when a virus does it, a virus hijacks your cells' machinery, and the cells then signal out saying that I'm hijacked, both by presenting the proteins that they have to express and by sending out signals.
Yeah.
What happens here? Does it send out similar signals?
It's your body reacting to the foreign RNA.
I thought it was to the spike protein.
What? No, that's the recruitment of dendritic cells.
What is the recruitment of dendritic cells? The mRNA?
They're reacting to the mRNA, yes.
But the mRNA is in the cells. I thought the mRNA was in the cells. So how is the...
It's foreign mRNA. Not all of it is going to be encapsulated in lipid nanoparticles. Some of it's going to be free-floating. Some of it's...
Oh, wow. Oh, this is all kinds of crazy shit that I didn't know about the vaccine.
Yeah, well, that's what happens in a tube. You don't have everything.
So then it doesn't stay in your shoulder either. It probably goes all through the body. It doesn't just stay in the shoulder.
It stays mostly at the injection site.
How could that be? I mean,
because you're injecting into muscle, not the bloodstream.
But that's not what happens all the time, then. If you
you're
hit a vein, or if you get close enough to an arteriole, then some of this stuff will pass into the blood, and then where does it go?
It's not going to go into an artery. It's getting injected into the muscle. It's very unlikely that any significant amount... How big is the muscle in a 5-year-old It's very unlikely that any significant amount of...
Okay, but let's say that you injected 100 million people. What happened? Would it happen in 10,000 people then, or what?
Hold on. It's very unlikely that any significant amount is going to ever make it into the bloodstream. There have been biodistribution studies to show that that's what happens in animals.
Okay, so it does happen in animals.
What? No, what I just said happens in animals.
What, that it stays in the muscle?
Mostly, yes.
What about that biodistribution study from Japan? That was bullshit then?
Nope. Less than 1% of the lipid nanoparticle signal went anywhere but the injection site.
In that study, they showed that like 25% after 48 hours was in the ovary.
Nope. That is not what it showed. Relative to the total amount injected, it was less than 1% that went anywhere else.
Interesting. And you don't think that matters if it goes to the brain or to the lungs or to the liver or to the heart or anywhere else random?
They certainly didn't find any evidence that it matters.
In those animals?
Yeah. No tissue damage whatsoever.
I see. What about that paper that recently came out where they showed that when it does go IV, it goes right to the heart and it expresses in lots of places. So it could occasionally. So if we injected 100 million people and 10,000 times it actually accidentally missed and hit an artery, it would be a problem. I would agree.
You're not going to do that in injecting it into muscle.
But it does happen. I mean, that
Can you
if
show
it
that?
happens in one out of sure. There's all kinds of forums where people are having all kinds of weird reactions to the vaccine that's supposed to be safe. Are those people all lying?
Show me the data.
Show you the data. You just have to go and find one of these groups on Facebook. There's thousands of people who claim to be damaged. Are they all lying?
Facebook's not data, dude.
Wow.
It's
Okay,
not. Facebook's
so-
not data. That's like treating VAERS as data.
What about VAERS? VAERS isn't data either, even though a doctor has to submit each of those deaths.
A doctor doesn't submit each one.
Of the deaths, they do.
No.
The doctor has to submit a death.
A doctor is not going to submit all of the deaths that are entered into VAERS. Sometimes it's a lawyer, sometimes it's a random person.
I see. So your belief is that the vast majority of those are crazy people, and so then we can ignore those 17,000 deaths?
No.
Is it just in America right now?
No, my belief is that you cannot trust the data at face value. You have to follow up on
Okay,
it.
so then let's say that it was a tenth. Would that be okay with you?
A tenth of what? The total reported
17,000?
deaths?
Yeah.
Well, that's not
17,000.
that's not what's shown.
What do you mean what's shown? 17,000 people have reported to VAERS and over 18,000 in Europe.
Yeah,
so
and
let's say
yeah,
it's
and a lot of
tenth
those VAERS reports are people saying that they're becoming the Incredible Hulk after vaccination.
Yeah, you can pick out an anecdotal joke like that, and then
I mean, you can
discount
pick out
the
an
whole
anecdotal
thing.
death like that too. It
It's
goes
not
both ways.
There are 17,000 anecdotal deaths then.
Yeah, they're anecdotes. You have to follow up and
Wow.
confirm them. You have to confirm that
Wow,
they
dude.
actually happened.
Wow. Okay. So I'm sorry, but at this time, I'm going to have to say that we're not really making any progress. I mean, you're definitely not interested in anything alternative other than the TV. And I can clearly see.
Oh boy.
So I don't know what else to say. I mean,
there's lots more we could get to.
Whatever. Please let me know. I mean, I just feel like we're kind of spinning our wheels here because you just say no and I just say yes, and we keep talking.
Well, yeah.
I mean, you tried to prove to me that the mRNA, you understood how the mRNA worked, and then you just kind of didn't say anything.
Well, yeah. You're kind of yelling at me, and we're jumping all over the place. But I'll show on my screen right here what I was talking about. So right here, strikingly, purification by FPLC has been shown to increase protein production from IVT mRNA by up to 1,000-fold in primary human dendritic cells. Thus, appropriate purification.
You're saying purifying it makes it more immunogenic, but that doesn't make it a more effective as a vaccine.
That's what a adjuvant does.
But
it's
that's
immunogenic.
the point, right? That's the point. So how does that jive with the idea that Moderna and Pfizer told us that the mRNA goes in and it expresses a spike protein, and then the spike protein, we make antibodies to it? How does the mRNA being an adjuvant figure into that? What does the body do with the mRNA then?
Are you asking how the mRNA increases the immune response?
If it does, what does it do? It signals it, like, you know, when you put an adjuvant in a normal vaccine, it's like a chemical, and it's a chemical irritant. And so they show up at the site. Listen,
okay,
they show up at the site, and then they pick up the antigen that you put in with the adjuvant. So in this case, you're injecting the mRNA, and some of the mRNA is expressing spike, and then the rest of it's attracting the immune system, and then the immune system shows up and pays attention to the spike.
Yeah, so you inject the mRNA. Some of it's in lipid nanoparticles, some of it's free-floating. The free-floating mRNA signals to the body that there's something foreign at that site. It recruits dendritic cells to that area. Once those cells in that area start expressing spike protein, the immune cells are there, ready to encounter it.
And so you think that that's the time frame. So you inject the mRNA, and then the mRNA irritates the immune system, and at the same time, some of the mRNA, some unknown amount, is entering muscle cells and expressing spike protein, and then a certain amount of time later, there's spike protein waiting for the dendritic cells that mature and show up there. That's what you think?
That's what happens, yes.
That's what they tell you happens in that.
That's what happens, because we
paper.
see a robust immune response from these mRNA
You
vaccines.
see a robust immune response in the sense of you see antibodies to the spike protein.
And cellular immunity.
What does that mean to you?
T cells. And
T
B
cells
cells.
to the spike protein.
T cell immunity and yes.
To the spike protein.
Mm-hmm.
And B cell immunity to the spike protein.
Mm-hmm. Yep.
And so
that's shown
polyclonal immunity to you means multiple antibodies to multiple different sites on the spike protein.
That's right.
And so that's completely useless because only the antibodies that bind to the receptor binding domain will block and neutralize the spike protein, and the rest do nothing.
No, that's not true at all. There's many ways an antibody can...
What do the rest do then?
They can opsonize, for one.
And they can't opsonize unless they bind to a membrane. They can't do that if they bind to a protein. Then they can only aggregate.
They can't opsonize unless they bind to a membrane.
They don't work as an opsonin unless they're bound to a membrane. But you would know that. You should know that.
Spike proteins are membrane proteins.
Right, but when the antibody binds to the spike protein, they're bound to a glycoprotein, not to a membrane. And so that's a very different kind of signaling. And in fact, that makes the part of the receptor of the antibody unavailable physically because it's in between other spike proteins. And that's the reason why a lot of these off-target non-receptor binding domain antibodies are responsible for antibody-dependent enhancement, like in dengue fever, which are also off-target glycoprotein antibodies made to last year's virus. But of course, you know that, right?
Yeah, dengue experienced antibody-dependent
But you know the mechanism, right? You know the mechanism is off-target antibodies to the glycoprotein,
because
the exact same antibodies you're describing now.
There were 2 strains of dengue.
And you don't think there are going to be 2 strains of this? There are many strains of this.
It's not a one-to-one comparison. You can't just say that.
It's not a one-to-one comparison, but the mechanism is identical.
What? No, it's not identical for dengue and SARS-CoV-2. They're different.
Off-target antibodies to the glycoprotein that allow the antibodies to allow the virus to enter other cells that it couldn't otherwise enter.
Okay, so are you saying that you're worried about antibody-dependent enhancement, or that we're already seeing it?
I don't know. I'm not in a clinic. I'm not running tests. I'm telling you that that's the possibility here are one of the possibilities that we're talking about. And the reason why making antibodies only to the spike and calling that polyclonal is incorrect. Polyclonal antibodies are to the N protein, to the M protein, to the E protein, to the spike protein. There are hundreds of papers demonstrating this from all the previous viruses, dude. Come on.
A monoclonal antibody is one antibody from one B cell. You have multiple B cells producing multiple different antibodies that recognize the spike protein in this case.
Yeah. And if they all recognize the receptor binding domain, that's still only one major epitope.
They don't all recognize the receptor binding domain. They recognize multiple.
But those are the only ones that matter. Those are the only ones that they measure.
No, they're not.
Okay,
no, it's not. It's not the only ones. It's not the only ones.
I guess you don't really understand that.
They don't have to bind Look, they don't have to bind directly to the RBD to neutralize. Even they can bind.
Yes, they do.
They can bind. No. They can bind adjacent to it and then sterically hinder it.
If that's possible, yes, that's true. That's an option. But then you're still not talking the way that you should be, which is that there are a limited number of targets on the spike that actually neutralize it, and the rest don't. And you keep backpedaling, but not admitting this.
I mean, this has been well characterized in studies such as this that I'm showing on this screen. I'll link it in the description for anyone who wants to read it. Lots of different antibodies, all able to neutralize and recognize spike, or otherwise deal with it.
You're still focused on the same thing that Moderna and Pfizer would like you to be focused on: spike protein antibodies. You really imagine that that's the mechanism of immunity, and it's not.
Well, of course, that's not all of it, but
it's not all of it. It has to have CD4 and CD8 positive cells.
Which
you have
it
to have
does.
CD4 positive cells coordinating the affinity enrichment of the B cells. You can't have the antibodies and the T cells are already singing.
Which it does. There is robust T cell immunity in response to these vaccines.
Robust T cell immunity to the one protein that changes the most in every coronavirus, the spike protein.
Which isn't an issue. It's not really an issue for T cells.
Oh my God. Oh my gosh.
Are you just going to keep saying, oh my God, or are you going to say stuff?
Oh, I am saying things. You're just ignoring it, right? I keep trying to explain to you.
You're not really saying much. So, what you don't think that this vaccine is going to produce robust cellular immunity against?
It doesn't. That's why people get infected more than once. That's why people are getting infected.
Is that why?
With a more severe case after what's happening in Vermont and Massachusetts, Israel, Iceland, what's happening in those places? Why are those people, what's happening in the UK?
You're seeing cases go up but no deaths. You're not seeing deaths go up.
I'm just
look at the UK. Look at the UK graphs.
It's not about deaths going up. It's about who's dying. The vaccinated people are now the preponderance of people dying in the UK.
That's
the last 6 weeks. 75% of the deaths.
That's not entirely true.
It's not entirely true.
Not really. No. So you have two things going on in the UK. You have a lot of cases, yes, but you have very, very low deaths. The low deaths are thanks to vaccination. Now, the deaths that are happening, because there's so many people vaccinated, a good portion of those deaths are going to be in the vaccinated. But if you break it down by age,
is that how it works with measles virus? The more you may vaccinate, then the more people get measles or are having vaccination. Is that how it works with measles?
Hang on. If you break it down by age, you see that the lower age groups that are less vaccinated have more COVID cases and more COVID deaths than in total.
And he explained that, Carrot explained that to you, and that's because these people with a non-sterilizing vaccination can carry higher viral loads without symptoms, and therefore they can infect the people that are standing around them much easier. That's an easy explanation.
That doesn't check out, really.
Of course it does.
No, it
they
doesn't.
have less symptoms. You say it yourself. The vaccines keep people out of the hospital, but they have the same viral load. So I guess then they produce. What are these studies that I showed in my live stream that are they? Lies?
I know. So okay, let's get to that. So you're talking about
the Wisconsin study.
Right. So tell me, what did it measure?
Do I have to get it up right now?
Just from memory, you should know, right? That's what you told me. You should know.
Sure. Yeah.
You should know.
That's very sweet.
No. Seriously, take your time, get it up, and let me know, what did they measure?
They looked at viral load, as far as I know, PCR count.
That's not viral load. That's the important point to make.
Yeah, that's exactly right. So how does that work for your favor? How does that work for your favor? That they measured viral load as PCR count. Who's doing it any other way?
Well, not many people, which is the problem. So viral load is not a PCR count. That's viral RNA.
But that's all we got. That's what everybody's doing.
Listen, that's viral RNA copy number.
You don't have to explain it to me. I understand it. I did it.
Well then, but you're calling it viral load.
Because that's what they call it, and everyone else calls it like you. The TV calls it viral load.
That's wrong. It's not viral load.
That's fine. So call it count then. How come they have the same count? How come they have the same cycle count?
Because there are lots of viral RNA copy numbers, but they're not necessarily shedding infectious virus.
Oh, and so that's what you can read from their data? That's how you interpret their data with your immunopathology?
Because
you have to take the data at face value, and there is no difference between those two groups in that paper. How can you differentiate between them at just imagining?
Listen. So, in your paper, they are measuring viral RNA copy number at one time point. Then they go on and they measure infectious virus, but they don't really measure it really well because it's just a yes or no answer. They say, does it have...
It's not suitable for you.
No, it's not suitable in general. So let me finish my point.
What about the paper that you showed where it has decrease down to you, but you just ignore the fact that the ones that are still carrying a load actually carry a pretty stable load the whole time, but you just ignore those?
No, hold on. So in your paper, again, viral RNA copy number, that's not viral.
There was one from
hey, hold on.
California.
Can I
got a debunk also, right?
Can I just...
Sure, try, I guess.
I'm trying. You keep talking, but it's viral RNA copy number in that paper. Then they try to measure infectious virus by just saying, does it cause cytopathic effects in culture, yes or no? That's not really good enough if you want to actually compare two different groups. So you can't really make conclusions that one group is spreading equal...
But you would make conclusions if the conclusions were ones that you enjoy.
No.
You just don't want to make conclusions because they are not convenient for you.
No, I'll tell you what I want to see if you let me finish. So the problem with that paper is you can't conclude that vaccinated, unvaccinated are equally spreading infectious virus. Now, what you would want to do is do either Well, I'll ask you, which two experiments would you want to do in order to determine that, to compare those two groups?
I want symptomology and only that.
Symptomology.
Yeah, I think if you're not
Those aren't two experiments.
I think if you're not showing symptoms, then we're all talking about mythology. I think that's one of the problems.
What?
That's one of the problems with this is that you like to use the PCR test when it's good for you, and then when it's not, you want to be instructive and say it's not viral load. It's
not viral load. I've never said it's viral load.
I get it, dude. I get it.
Okay, so what two experiments?
What two experiments would you do? Why don't you be the smart guy?
I'm asking you. So it's okay. It's okay if you don't know.
Yeah, it's okay. I don't know. I want you to be the genius. What two experiments would you do?
Right. So you would need to do either a plaque assay or a TCID50 in order to determine what amounts of infectious virus either group is spreading, so that you can compare. But that
what plaque assay would you do?
What do you mean? What plaque assay?
Is there a standard one?
A plaque assay is a plaque assay.
It's not. You have to have a background. You have to have a positive and negative control. What are you talking about? You're just pulling something out to try and look smart, right?
What are you talking? No, a plaque assay is determining the amount of infectious virus. How would you decide whether it was one of the 4
endemic coronaviruses or this one? With sequencing then after you do the plaque assay or before?
Yeah, you would determine that
because these PCR counts, none of them were sequenced. We don't even know for sure if these were any of the 4 original coronaviruses or this one, because we don't even know what 3 targets they sequenced for in this one. Right?
Are you saying that?
I'm saying that at least half of the PCR tests, unless you sequence afterward, cannot differentiate between the 4 endemic coronaviruses and this one. And that's been shown. That we know that.
No, that's not true.
It is true, though.
No, it's. Oh my.
The WHO even admits it, dude. Why are you
what do you mean
not knowing this?
Okay, hold on. What do you mean the WHO admits it?
The WHO's policy admits that you have to sequence after you get a positive, or you cannot determine whether it's SARS-CoV-2 or not. That's already been like a thing for a year.
That is not true. PCR.
The CDC also says it. The FDA also says it, dude. It's in the EUA. You haven't even read it.
That is entirely not true. So I have several videos talking about PCR.
I'm sure you have videos talking about it, but you've got to read the documents, dude. Otherwise, you're just reading television and regurgitating what you saw on television.
I don't watch television, dude, so it's weird that you keep saying that. Okay, the qPCR for SARS-CoV-2 is specific for SARS-CoV-2. It was tested, there's an FDA document that I link in several of my videos, where they go through and test it against all of the known human coronaviruses, SARS coronavirus, influenza, the gamut. And it only comes up positive with SARS-CoV-2. That's the power of PCR, is being able to differentiate between such closely related species.
And so you're convinced that all 246 PCR tests that were given an EUA by the United States in 2020 were all using the same primer sets?
Yeah, I mean, well, no, they're not using the same.
No, they weren't, though, because the WHO had their own primer sets, dude.
They're all specific. They're all validated in-house in every clinical lab.
That's the assumption you would make, right?
No, that's how the industry works. You have to validate each test in-house. It's very robust.
Very robust.
Mm-hmm.
That's why they went out to how many cycles did they use in the beginning for the first year? 45 or 40? 40, was it 40? 42? Wasn't even 45? It
doesn't matter.
It does matter if you know how PCR works, dude.
I know very well how PCR works. So how about you tell me how it works first, and then I'll respond.
Well, PCR is amplifying the presence of any DNA, right? So first you have to convert whatever RNA is present to DNA, and you only get one chance to do that. So whatever RNA is there is going to get transcribed to DNA. I don't know the right word for that, reverse transcribed to DNA. And so whatever RNA is available, it could be endogenous RNA, it could be bacterial RNA, it could be viral RNA from any number of rhinoviruses, adenoviruses, whatever viruses are present in the sample you take. This is very different than the PCR that you and I do in an academic laboratory, where we take a very, very fine sample, and it's very, very controlled only for the thing that we want to amplify so that we can prove its existence. I've done reverse transcriptase quantitative PCR to prove the existence of particular nicotinic receptors in individual neurons that I pulled out of the brain. And when I did that, I had to have positive controls for different subtypes of the receptor. I had to show that my primers all work for those different subtypes. And for a lot of these tests, those have simply not been done.
Yes, they have.
A lot of these tests do not have a positive control, for example, for any endogenous RNA. They don't have any controls for any regular everyday coronaviruses that should be present in some of these samples. And so as a result, whether you like it or not, there is a possibility for there to be false positives in light of the fact that we are testing people who are asymptomatic.
Okay.
Because testing on a low prevalence background is a very, very, very bad thing.
It's not, because qPCR is so precise. So the way that it works in this case, you were talking about cycles, and that's pretty concerning that you're talking about it that way. I didn't think you would. I didn't think you would go there.
Explain it to me why that's concerning.
Right.
So I don't think it really is.
qPCR in an industry setting, in a clinical diagnostic laboratory, you're going to have a set of standards which act as your positive control. These standards are going to be serially diluted, and they're going to set a range, right? So, for example, if you're running a test for 40 cycles, and your standards set a range of 12 to 33. Any samples within that range are positive. Any samples outside of that range are going to be deemed either undetermined or negative. So it doesn't matter. That's why it doesn't matter how many cycles you run it for. The cycles are determined based on the standards you have and the platform you have and the specific way your lab is setting up the test.
And so the cycles cannot be used at all. What does the Q stand for then?
I didn't say the cycles aren't used at all. I said that you can run the test for 60 cycles if you want. The range is still going to be the same. You're not.
I understand. So if the range of your indicator of being reasonable is between cycles 8 and 32, and they were using cycles up to 45, you have to admit that then those are all false positives, or many of them will be.
No. What do you mean they'd be false positives if they run the test for 45 cycles?
Because you're building up a fluorescent signal in your commercial test. Your commercial test is not an academic like yourself doing all the necessary controls and looking very carefully to see that the sample is reading the way it should be. It is a machine beeping at a certain number, saying that the fluorescence is at a certain thing, and they don't sequence it, they don't know. And so if you have a mRNA that was significantly overlapping enough to transcribe, you're going to get a positive in one of those three. And if they only make a positive in 2 out of 3 to make a positive, then you don't have a result. And they did this for millions of people without people trained as you are trained, without the skills that you have. These are technicians at best doing it as fast as they can to make as much money as they can. That's what's been happening in all the track and trace things that have been going on for a year and a half, because that's what all these private companies have been doing.
Okay, so you're saying something that's making me concerned again. How many measurements are taken during a qPCR experiment?
For which? SARS-CoV-2? I don't know. It depends on which test they do.
Any qPCR, how many measurements are being taken?
When I did it, I'm amplified like 20 different transcripts. I tried to do single cell PCR and amplified many different transcripts to see if they're there. So I was doing 20 experiments at once using 20 different primer sets.
So let me try to figure out how to ask this a little more clearly. If you're running
you're a molecular biologist. I'm a neurobiologist, so there's going to be things that, molecularly speaking, you're going to be able to make me look like a child, and that's fine. But you're not making me feel any better about your immunology or mine. I mean, I don't understand.
Okay. So if you have one sample and you run it in a qPCR test that runs for 40 cycles, how many measurements are you taking?
Presumably one.
You take 40 measurements.
Sure, you take 40 measurements, but at the end you have one sample. You don't have 40
No.
samples.
No, so qPCR is done in real time.
Right, and it's in the same tube.
It takes a measurement after each cycle.
But at the end, you're done. You have a pile of
But you
your
don't just
product,
you
right?
don't just measure that pile. You don't just measure the pile. You measure after every cycle.
How the fluorescence changes per cycle. Sure, you get
Yes,
a curve.
exactly.
You get a curve that describes the change in fluorescence.
Exactly, and when the curve passes the threshold to being positive, that's what's recorded as the cycle threshold positive. So it doesn't matter if you run it.
What are you doing? Is this like a gotcha moment where you're trying to get me to explain how PCR works, and then I explained it, and now you got me? What is this?
You brought up PCR and started saying things about it that told me you're not really understanding it. That PCR is.
But I just explained it to you. Of course I understand it.
You didn't answer my questions very well, and you're talking
Because I'm
about
trying to explain
you're talking
it to
about
other
SARS.
people.
You're talking about SARS-CoV-2 PCRs not being specific for SARS-CoV-2, and that's not correct. And you're not understanding.
It's not correct in your mind, but I mean,
it's not correct in reality.
I can. Well, after the stream, I would like to connect you with some actual other people that are doing this argument, that are molecular biologists making the argument that many of these.
Oh yeah, I'm in contact with a Dr. Lee. We're gonna hopefully do one of these. The whole false positive narrative about these COVID PCRs is totally wrong. If it were,
I'm excited. That's good. I'll just let
if there
him do
were
that for you.
if there were
because
rampant if there were rampant false positives, we'd see very high PCR rates going consistently throughout the pandemic. But
what? Why would we see that?
Because if that's what we would see if it was rampant false positives detecting endemic coronaviruses.
No, but they're not. They don't have to detect anything. They just have to produce fluorescence in the tube. That's the point of the commercial test.
But
they
only detect what produces fluorescence, and the fluorescence is based on the sequence.
Right. So if you have any primer dimers or anything else that could possibly produce a false positive, it's there. And if you've done enough PCR, you know that false positives happen, and you can't always explain them by the PCR is just so good, and my primers were so good. How can this be here?
I do qPCR a lot.
On very clean samples, not from tubes that you take out of a nose.
No, I do it on crude extracts, and I have almost never seen a false positive. Actually, none of my negatives have ever turned up positive. It's just not common. It's extremely uncommon to see false positives. It's uncommon,
but we're talking about hundreds of millions of asymptomatic people that were scanned for a protein that they probably didn't have, but might have had protein in the past. So how would you said it yourself, right?
Protein.
People with mRNA. RNA. If they have RNA fragments from viral infection that are still there from weeks or months ago, whatever you said earlier, that vaccinated people can have RNA in their nose, but it's not infectious, and you have to do a plaque assay to figure it out. So then all of these asymptomatic people that were PCR could be put in exactly the same category as having mRNA that's not infectious, that was from previous infections, that is coming up positive, and now you're shaking your head as though previous Mr. Wilson was wrong.
That's not how it works. You're not. It's really uncommon to detect previously infected mRNA in a person.
Then how are you using that as an argument to say that those viral loads in those vaccinated people aren't the same as in the unvaccinated people? That was the exact same argument you used.
What? No. Okay, let's get back to the...
No, you used the same argument. You said that you have to do a plaque assay on the vaccinated people in order to prove that the PCR positive that they're getting is infectious virus. But now you're saying that there's no possibility of there being uninfectious virus in people.
What? No, I didn't say that. What are you talking about?
Oh my God, dude, you're lost.
Okay.
You are lost.
Right.
I don't think
so. So let's get back to
my kids to bed. If you want to do this again, we can do it again. But it's not really. This is not really doing anything. This is just.
All right. Okay.
I'm giving up. I think you win. I hope that in a year we'll come back and talk to each other, and you'll still have the guts to talk to me.
Hang on. I just before you go, I just want to say that let me sum up my point that I was trying to make. So, yeah, you can't just take PCR copy number and say that that's viral load. You have to do a plaque assay or TCR50. But all of these viral RNA copy number experiments, including the Singapore study, show that over time, vaccinated people clear the virus faster. This indicates that a vaccinated person would transmit less than an unvaccinated person. And if we look at epidemiological data, like what I have on my screen right now, that absolutely checks out. We see a very significant reduction of transmission from vaccinated people relative to unvaccinated people.
And the reduction in transmission is not to zero, and that's the point that Geert is trying to make, that if you don't have a sterilizing vaccine,
it doesn't need to be zero.
Can you listen for a second? Then the vaccine, that the viruses that these people are transmitting are viruses which thrive in the presence of the antibodies that you're so happy with. And that means everybody with this complement of antibodies will be welcoming of the viruses that these people do transmit. So even if they don't transmit for as long, the viruses that they are selecting for are the dangerous ones. And that's why Trevor Bedford's discoveries on next strain are so important, and you have to ignore them. Because the only proteins that are changing are the ones that go into circulation after transfection. And that's because then the immune system makes an overabundance of redundant memory to that protein, and that's why it's forced to change. You're going to see it. The hospitals in the United States will be full of vaccinated people in less than half a year, and you'll have to explain it with something other than the stuff you're talking about now.
Sure, we'll see if that prediction comes true, but let's see where to start. So you don't need to have transmission reduced to zero. That is not a goal of a vaccine. Nothing can reduce transmission to zero.
Measles? That doesn't happen with measles or with polio? They don't care about that?
Polio vaccines actually do not prevent infection.
Is that true?
Yeah. They prevent paralysis and disease, but they work really well. They work really well.
These don't, though.
They do. They do work really well. So what's going to happen in the case of an mRNA vaccinated person getting infected with SARS-CoV-2 is the virus is going to infect, it will start replicating in the host, because vaccines do not prevent infection, they prevent disease. So if their antibody titers are not super high so that they can neutralize the virus before it can really begin infecting, then that's what you're going to have. You're going to have an infection. But as it begins replicating, the immune system will mount its response. Before the immune system actually mounts its response to clear the virus RNA copy number is going to get to a certain level. That is mostly not going to be selected for by your immune system. So if a vaccinated person transmits, which is possible, it's likely going to be in that period before the immune system has really responded and started neutralizing and clearing virus. And unless we have experiments to show that vaccinated people are in fact selecting for infectious variants that are being affected by natural selection here, then you can't say that. You would need data to say that, but there is no data to say that.
Except for data that shows that the variants of concern are predominantly in vaccinated people, whereas the unvaccinated people have very random viruses. That sequence data is what is in Trevor Bedford's papers, but that's okay. You don't bother.
I mean, you could show it. You could show it to me.
I did it on my stream. The links are there. Like, dude, you're so far from wanting to learn, and it's really it's too much friction. It's not a lot of fun.
Okay. You seem very upset, but I mean, the next strain data.
I'm upset because my kids are about to be transfected against my will because of dipshits like you thinking that transfection is the same as vaccination, and
it is a vaccine. It elicits an immune response. That's what it does. It's better described as a vaccine than it is a transfection. You're just trying to scare people.
Transfection is exactly what we've called it for 20 years when we've used mRNA to express proteins in other animals. Don't be a dumbass.
Yeah, but it's better described as a vaccine.
Yeah, for the kids at home, because then we can indemnify all of the companies that are producing vaccines under the law that protects people that make vaccines. Dr. Wilson, thank you very much for joining me.
Wow,
it's been really great.
Well, I'm sorry it couldn't be more civil. He's gone. Well, that's me and my wife. Let me get my camera set up here. Is that working? All right, my webcam's not working. It looks like he rage quit. That's fine. I didn't expect this to be... I didn't really expect this to be as much of a dumpster fire as it was. That was weird. Why is my camera not working? All right. That's okay. I'll show data. How about that? So, this is actually, I'll just, yeah. So this is the Singapore study that he talked about in his stream. He didn't really like it much. All right. Let's see. Sorry, I'm reading messages here. Where's this figure? Right, here. So he didn't really like this figure. What this figure is showing is it's showing viral RNA copy number in unvaccinated versus vaccine breakthrough cases. And it's showing serial CT values over time. So this means that these are the same people followed with the same test over time. And over time, you can see that on average, vaccinated people and unvaccinated people start out at similar RNA copy numbers, but then over time, vaccinated people clear faster. And if I remember right on Jay's stream, he said that this is bad because they're factoring in all these zeros to the vaccinated people, which is bringing the curve down. But hopefully you all can see the problem there. These are serial CT values. So these people are clearing the virus and going to zero. They're not starting at zero. So they're getting to zero faster than the unvaccinated people. So that data is nice. It would be nice if we could have infectious virus. It's driving me crazy that no one's doing that experiment. That's not the right paper. Which one is it? This paper actually just came out in The Lancet, which is showing data that is consistent with the Singapore study. Basically that vaccinated people clear the virus, including Delta, faster than unvaccinated people. Right? So that's all well and good. Let's see. I guess we talked about most things that I kind of wanted to talk about. I didn't expect him to bring up the PCR stuff. I didn't expect him to subscribe to that, but that's pretty telling. And again, here's that mRNA vaccine review paper where they talk about the things they did to make mRNA more immunogenic while still making it stable and not be too immunogenic. It's a really interesting read in general. If you're hesitant about mRNA vaccines, I highly suggest you read it. And it actually looks like, yeah, hold on. Let me get a guest on. Let's see. I'm just gonna get a guest on a Zoom call right now. And that's my wife's name. All right. Oh, I changed my own name. Anyway. So one of my friends, it looks like, wants to get on and let's get him on here because that was an interesting conversation. Yeah, I really wasn't expecting him to bring up the PCR stuff. That's really bad. If there were rampant false positives, you would expect there to be a lot of a really high and consistently high percent positive count in countries across the world, but you don't see that. In countries that actually control the pandemic, like Australia, for example, the percent positive, and percent, when I say percent positive, I mean the total number of people who test positive within your test population. That remains pretty low. Sorry, I'm trying to click and talk at the same time, so. All right, there we go. Okay, let's look at the chat. Oh, I see Tariq's here. Yeah, they're covering up the false positives. Yeah, that's a good point. Multiplex PCRs would be showing false positives, but they aren't. Yeah, it's. I was pretty surprised by that. Yeah, he was pretty upset. But I guess that was first rage quit. Interesting. I really tried to keep it civil. Let me know if there are better ways I can do that. But yeah, he seemed to get really upset really fast. Oh, I have to let you in. My bad. How do I change my name here? Hey man, how's it going?
Dr. Wilson, good to see you. Good evening. How are you doing? You had a good time there, I see.
Yeah, that was interesting.
Oh boy.
All right, so we got Senstrand here. He's been on the channel before. Yeah, that was a pretty interesting chat. We didn't really get through everything, but man, the PCR stuff. What was up with that?
I was pretty shocked. He was at least partially knowledgeable on the immune system function, and then really kind of showed his hand with PCR because, well, I guess as a neurobiologist, he said he was a neurobiologist.
Yeah.
Yeah, he's had exposure to it, but didn't understand the basis enough to really complete his thoughts on the PCR. So you did a good job trying to explain it to him, but he got frustrated at that point and left. I think he could have just, let you say, okay, this is how it works, and then made his points and then continued. But he was very aggressive from the onset.
Yeah. He emailed me that he said, I think his words were he was going to be relentless in trying to convert me, something like that.
That doesn't really open it up for a good discussion.
No.
In my opinion. I would like to have known what the hell he was talking about the variants when you're vaccinated, right?
Yeah. So as far as I understand, he wrote up like I guess, I don't know if you call it a paper, but he sent it to me. It's not published. His argument there was basically that as soon as the vaccines come online, you see this increase in variants on next strain. But really, that you can correlate that with the fact that people were ramping up testing for variants. That was around the time at the beginning of the year that people really started to become concerned about the UK variant, the South Africa variant. And people said, we need to start tracking this. And so a lot of efforts worldwide started going into actually sequencing PCR positives. So then, of course, you get a lot more data going into next strain. So
his exact words were, they've studied and shown that vaccinated people harbor more dangerous variants. And I don't know where he would have gotten that data from.
He said true.
And also, and also he's implicating that vaccines themselves are a part of the genesis of new variants and new more deadly variants, and that's not true. The most deadly variant that we're dealing with originate from countries that was my daughter. Apologize.
Yeah. Thankfully, mine's fast asleep. Yeah, I'm looking. He said Trevor Bedford. As far as I know, it's just like a correlation with next strain data.
Okay.
Yeah, here it is. Right. Yeah, it's just next strain tracking. I don't know of any data where someone actually measured vaccinated people selectively spreading different viruses.
Yeah, right.
Yeah.
So then he's misinterpreting that data.
It seems like.
My main point is he says you never bring up data, you never bring up papers, and then says, well, you bring up data and papers, but it doesn't have my interpretation, so.
He did that a couple times,
therefore
right?
You're frustrating me, and it's like, well, which is it, dude? Yeah, he did that a couple times.
Did that a couple times.
Also, you invited him to bring up data, and he turned you down at each venture.
Yeah, he did that a couple times where he said, well, this doesn't happen, and then I said, well, it does, and he said, well, yeah, it does, but blah blah blah.
Did you want to revisit what he was saying? I mean, he just needs to stay on track, for Christ's sakes. It's with the mRNA vaccines, and so we know that mRNA will induce an immunological response just for being mRNA, correct?
Yeah. Mm-hmm.
Yes. That response is not specific,
right,
and leads to degradation of the mRNA, right?
Yeah.
So then it ended up being a balancing act in creating these vaccines, which they are definitely vaccines. They're not transfections. I don't.
Yeah, I mean, yeah,
the vaccines induce an immunological response. That's the definition of what they are.
Yeah.
And the method of delivery doesn't play a part in that equation. The transfection that he's talking about is essentially a gene therapy that's looking at, that's helping to remedy mutations or diseases that are genetic diseases. So, right. So the backbone can have the same technology and have a completely different outcome.
Right. Yeah, it took a lot of work because at first they were, with the mRNA vaccines, it was tough to get the mRNA stable enough so that it actually did anything in the cell before it got chewed up. And then they had problems with delivery. They had to make the lipid nanoparticles less toxic. And that was all part of that's all described in that review. I'll link everything that I showed here in the description, but that was what the review talks about.
Dr.
Trying
Wilson, you never link papers.
I don't. I know. I'm sorry. I must have just forgotten in all of my videos to link all of the papers.
Right.
Let me go back and fix it right now.
I've just never once seen you cite PBS NewsHour. So,
yeah, that was an interesting.
hilarious.
that was an interesting choice to call all of the. I mean, he doesn't just do it with me. I listened to his stream where he talked about my video a little bit, and he was just calling lots of people TV doctors, TV scientists. I can't remember the last time I watched television.
Right. As a scientist, you really have no time for television. Honestly.
Yeah, I don't watch those press conferences unless like some conspiracy theorists are talking about them and they clip something and I have to watch it. But yeah, I don't watch those. I just go to work, read papers, listen to podcasts, come home, take care of my daughter, and then
Right.
and then try to do YouTube when I can.
Yeah. It take hours and hours just to read and go through a single paper and make sure you have all the bases down. But no, he really faltered and failed on the qPCR. Do you know what he was even trying to get to on qPCR with the viral load? First off, good job pointing it out that viral load is not equivalent to detected viral particles.
Mm-hmm.
Right,
Right.
you had to go through subsequential steps that he seemed a little bit confused on.
Right.
And if I'm not mistaken, was there not the paper that said like, well, the detected amount of viral particles was as high in vaccinated individuals who were symptomatic.
I don't remember actually if they made that distinction.
As far as I know, it was symptomatic individuals, but
Okay.
the quality of those particles were low.
Okay. Okay.
I'll have to pull up those.
Yeah,
studies.
I don't remember those details, but
Okay.
yeah, they had similar RNA copy number at the time points. I think it was like minus 3 days to plus 4 to 6 days. They have similar RNA copy number values.
But
But
purging much faster.
yeah, but the being cleared much faster. Yeah. But yeah, I'm not sure what he was. He tried to say something about, oh, you're using it here, but then using it differently here.
Yeah.
I don't know what he was getting at.
I'm trying to figure that point out too.
I think-
I don't quite.
I
I'll have
think
to rewatch. Mm-hmm.
I think I'm not sure that he was trying to say that mass testing people with PCR, and then like if someone comes up positive and you have to self-isolate and stuff, that he's mad that has to happen because they're not proven to be infectious. I think that's what
Right.
he's getting at,
Right.
but that's
Okay.
not really the point. Like, we can't do TCID 50 and plaque assays for every person who
you
becomes
would never be able to.
positive. That's what those assays are, super labor intensive. I do TCID 50. It takes like a week and a half to complete the assay. And so
You would never.
you would
control,
never, no.
but you would never have any any clue or any hint.
no.
It gets back to the confusion that I see people when they say this is a false positive, when they really mean that you have a low incidence, you have a low detection, and therefore you are not infectious. Therefore you're false positive. That's not what a false positive is.
Mm-hmm. Right. Yeah. Having a really high CT value, which is low copy number, that's not necessarily a false positive. It just means that, like, it could mean lots of things. Which is why if you get a positive, you're told to just be safe and self-isolate unless you want to do another test. Because it could mean that you're in the early stages of the infection and the viral RNA copy number is creeping up, so the virus is replicating. It could mean that you're at the end of an infection and the number is creeping low, or it could mean that you did end up getting a false positive somehow, which is again really rare.
Yeah,
So
it's really rare. I mean, it certainly happens, and it's just, well, on a personal level, how are you going to take? First off, I've had at this point, nasal pharyngeal tests because I'm exposed to students and people that are, potentially immunocompromised. That so I've had like 20 of these. Each of them have been negative. I'm more concerned about the rate of false negatives, to be honest.
which is higher than false positives.
Right.
Yeah.
And they're uncomfortable. Yeah.
Yeah.
I know a colleague that got just went in for like a routine operation, and they had to test him, and he had the sniffles but wasn't expressing any severe COVID symptoms, and he tested positive with rapid, and then tested positive again with PCR, right?
Yeah. Yeah, that's happening.
Yeah.
That's happening a lot now.
So he was like, I didn't even know. So he had a quarantine for 10 days at that point, and then we all had to get tested. And none of the people who were around him were spread in at least the office. His wife got it, but in the office, our preparations, we all always wore masks and maintained a decent social distance. So at least at that rate, and probably lowering the amount of viral copy number through vaccination, because he was vaccinated, probably spared at least a couple of us from getting that infection and spreading it again. Right. So that's another generation of infection spread. So
right. Yeah. I just saw So Clemens Load says, regarding false positives, his point was about the degrading probe producing a false signal. Does a standard machine recognize this? That signal would be linear as opposed to exponential. Yeah, it would be linear. Probe degradation, you always run a negative control of just blank in your assays. And usually you can see probe degradation. It usually will like creep up way at the end of the test, but if it does end up like passing the threshold, it's always outside of the range of the standards, so it will read as negative. There's just it's not really a concern.
Primer dimer as
Primer
well.
dimer, yeah. I mean that's not really gonna.
It's predictable and testable, and
right.
Again, each diagnostic, and he seems to say like things that I can't immediately falsify, which is or you can just be like, no, I have the data because you don't have preview to that data, but just saying that these labs are run differently is like, no, they have to be, they're regulated to run in a specific manner to be a diagnostic lab. Right, yeah. Which means you have to run in a certain amount of runs a standard control.
Yeah, and that was kind of a nasty low blow to say that these people are untrained. I mean,
they are definitely not untrained.
These are people who are doing like the same assays all the time in these clinical labs. They know how they work. I work with people who don't have PhDs. They're very smart people. They know how to run assays, and all of their work gets sent to review. So even if a technician, for example, someone you want to call a technician, I would call them a scientist, will run an assay, get their results, and then they'll have to send their results off to get reviewed before they get like finalized. And the reviewer is always going to be someone with a PhD.
Right.
So they'll be able to spot things like false positives coming from, like, for example, contamination in a well will be pretty obvious to spot because there won't be like everything's serially diluted. So if one well in the middle of a serodilution is like shooting up, that's a
Yep.
pretty clear indication of a false positive.
Right.
And they would say that that's not a real positive. Either rerun the test or report this as undetermined.
Right. Right. Exactly.
So-
Yeah. Okay. So Clemens again says, I think his concern was that the PCR machine just returns true or false.
That's what I was trying to get at, because I think a lot of misunderstanding around PCR is thinking like, the regular PCR, where you put the tube in a thermocycler, set it for however long the run is, you come back, and then you run a gel and you see how much DNA is there. But that's not what qPCR is. It's
Right.
taking a measurement every single cycle, so you're seeing it all in real time, and then you take that data and you interpret the data using your set statistics and analyses you have set for that assay that are developed and validated in-house.
So anyway,
Can you share what other viruses are detected using RT-PCR? Yeah, lots of viral diagnostics are used. People diagnose the flu vaccine. They've diagnosed flu infection with PCR tests. During the SARS-1 outbreak, they used PCR to diagnose those infections. It's really common in clinical diagnostics to use PCR. So it's not some new thing that is being used for COVID.
and the new approved test will multiplex flu and COVID.
Yeah.
Just to skip sampling and.
Yeah,
So,
it does flu and COVID at the same time. So it'll say either yes, it'll say positive for flu or positive for COVID, in the same test.
Oh yeah. Of course the Carrie Mullis
Yeah.
statement that is always we've addressed that multiple times. You have addressed it multiple times.
I have. Yeah.
Yeah, it came up again when we're talking Baby to Blind Joe.
Oh yeah, that'll be fun.
Yeah, he seems like a good guy. We'll see.
Did you
so
see
Templar, or go ahead.
Oh, sorry. Did you see my email about the hypnoses saying that the vaccine is a choice point and disrupts the connection to the heart? I haven't gone through my channel's email in a little bit. Sometimes I'm on and off with that. Sometimes I just go like a week without really reading the emails. So no, I haven't seen it yet. I'm assuming you want me to debunk it. That sounds wacky. So I'll check it out. But no, I haven't seen it yet.
I mean, it all gets down to like mistrust of the entire system, to the fact that like you're not even getting a vaccine. I don't know. I'm just spitballing off of what that last comment was. Like, you're not even getting a vaccine. You're just getting hypnotized to say that you've gotten vaccinated. So you mistrust the system so much. And I'm not saying it was Dr. Couey that you had on.
Dr. Couey.
No, I'm sorry. Who did you have on? He didn't even show his face, which is okay. But,
Oh, just now? Oh, Couey.
Yes.
Couey, I think. I don't know how to
Okay.
say it, but I think that's how it is.
He just mistrusts everyone, right? And a major part of his argument is that, well, he mistrusts Pfizer. He mistrusts data on vaccines. He
Right.
mistrusts the diagnostic clinic. Because, like, okay, you just listed hundreds of thousands of people that you just mistrust.
Right.
Who are professionals that do this all the time. This is what their jobs are, not just a sole company. And he can't point to a single piece of data point that would support any of that mistrust.
Right. It's just really like all of these worldviews require you to just reject a lot of people, which, it's not good to argue from authority and all that, but to assume that so many people are just lying, and most of which are experts, It's odd. It's an odd worldview to me.
It's a current state, unfortunately,
Yeah.
things. So,
So this is
um,
a that's a good question. How does sample collection standardize the starting quantity of sample to infer viral load? It seems a non-precise volume is collected at each collection. Yes, that's a very good point.
mm,
So for determining, so if you do it from a swab, that's probably not ideal. You'd probably want to do a nasal wash if you want to do actual viral load. But in these viral RNA copy number experiments, they are using swabs, I believe. And that's, of course, not a standardized way because you can get varying amounts of material on the swab depending on who you swab. So it's definitely, there are definitely caveats to it. It's not a perfect way to collect samples, but for trying to figure out if someone is infected or not, it's good enough. For determining amounts of infectious virus someone's shedding, it would probably be ideal to either to just collect multiple samples at multiple time points in the same day. But not many people are doing that work with SARS-CoV-2. No one's really determining infectious shedding of virus, at least not using the quantitative methods like plaque assay and TC-ID50. So we'll see. We'll see what the data say and how they do the experiments, because they'll do it eventually. They have to. I would hope.
yeah, I agree. They're going to have to do those experiments just to really prove their points on it. So, let's see. Bad quality samples are going to affect detected genetic material. Sample quality will,
Yeah.
will, yeah,
Yeah.
yeah. Again,
some
more likely causing a negative than a false
yeah,
positive.
because you'll lose DNA, or sorry, you'll lose RNA in the sample. So yeah, bad sample handling has been historically an issue in isolated labs. So there's isolated incidents. You can find examples with COVID and other diseases where they find that a batch of samples weren't handled correctly or something or other, and so sometimes it turns into a story of all these samples are giving bad data, and sometimes it's spun as like PCR is not working, but really it's just stuff happens. People make mistakes, samples get mishandled, and work has to be redone. It doesn't mean the test itself doesn't work.
Yeah. Let's see what else you got going on here. So, of course, it all came down to him basically just yelling at you while you're trying to pull up the information, which is not a fair way to do a discussion.
No.
I think the whole thing again. I don't know if we said it very explicitly. The whole thing about adjuvants and mRNA. When you were reading that, there are other compounds that can act as adjuvants in the vaccine, correct?
Yeah, so in the paper I started reading a passage where they talk about adding adjuvants to mRNA vaccines.
Right.
But it's a review of the history, so they go on to explain how they found out that mRNA could act as its own adjuvant, which is good because you'd expect that to be a good thing with anti-vaxxers because the adjuvant is a lot of times what they get hung up on. They say, I don't like aluminum in my shots. I don't like what are the there's another adjuvant they recently harped on, but I can't remember the name. But they usually hate it. They usually say that adjuvants are poison or something or other. So you would expect that to be a good thing, but apparently to him it means it's not a vaccine.
Yeah, I don't know. It's very Very interesting.
Yeah, I was
Well,
hoping.
maybe.
Oh, sorry, go ahead.
Yeah. Well, do you think he'll reconnect and try to get back on? Or you think he's just gonna? I
I don't know.
mean, at least if he does, tell him like you can't just storm rage off like this. It's not a fair way to conduct a discussion. And yeah, I'm totally. I mean, you're totally willing to change your mind or learn if he presents data.
Totally, totally, yeah. If
But again, he
he,
presented none of it. No, none of his points had data. He just kept saying like, "Well, I have data, and that data is in my video, so go see my video."
yeah, I mean, if he showed me data that showed that vaccinated people shed infectious virus that is evolving toward, I guess, more pathogenicity, more immune escape, whatever, then he might have a point. Then I would acknowledge that as data, and we'd have to probably the solution would be to redesign vaccines or roll out some version of a solution, whether it be boosters or new next generation vaccines. But that data would be evidence of what he is saying, so I would totally acknowledge that.
Right.
Yeah, I was hoping we would get to like more about the cellular immunity, because one thing he said when he was watching my video, I start talking about how when you get vaccinated and you have this immune response, your T cells are going to be able to recognize the spike protein in ways that are different from antibodies. And that's going to contribute to these really, really low chances of vaccines actually escaping, or sorry, the virus actually
Oh,
escaping
okay.
vaccine immunity,
Right.
because from person to person it's going to be different. And he said something along the lines of, "Oh, this is the limits of his knowledge. I don't think he knows what he's talking about." And I wanted to ask him, do you know that I'm talking about HLA genes, which code for MHC, which is going to
Hmm.
be different from person to person, and MHC is what presents the peptide to the T cell, and that's what the T cell is going to learn to recognize. So MHC is a little bit different between people to people, so the peptide it binds is going to be a little bit different. So the T cell immunity is going to be a little bit different.
Yeah.
So
Yeah.
even if a virus in a vaccinated person learns how to escape their T cell immunity, it's most likely not going to help them much when it moves on to the next person, because
Right. Because that coding is different.
yeah. And so for anyone listening who might not really doesn't really make sense to them, just think about how many people are organ donor matches to each other. It's pretty rare, and that has to do with your HLA genes. If they aren't as good a match, if they aren't a very good match with each other, then your immune system's going to attack the donated organ, and that's a big
Yeah.
problem. So yeah, that's what I was talking about, and I don't know if he understood that, because it definitely goes against Hertz's point that vaccines are somehow going to make the virus evolve faster and escape immunity, and we're going to have this huge problem on our hands.
Yeah.
It's just baseless.
Well, he threw it out there, so it's like you just have to wait a half a year and see if the hospitals are filled up with, which honestly it just doesn't make any sense. But he threw out the prediction that the hospitals are going to be filled with COVID patients, but they'll only be people who got the vaccination, which doesn't make sense. Why would that? Why would the vaccination cause you to become more susceptible to the virus?
Yeah, I don't know.
But not people who are unvaccinated. So
Yeah,
the virus has evolved now to escape the vaccine, and not only escape the vaccine, but become more infective to people who are vaccinated. Is what he's saying for his prediction to come true. And that's not. First off, it's going to be difficult for the virus to overcome the structural changes needed in the spike protein. That's why the spike protein was chosen, because it is at least conserved enough that if it changes its structure enough, then it might not get recognized by the vaccine, but then it will also not gain access into the cell. So,
Yeah.
I don't know.
Yeah. It's pretty weird. This person says it's giving me it's a YouTube video of a seminar from Trevor Bedford. Okay, I'll check that out.
Who's Trevor Bedford anyway?
He is the guy who, he's the guy that Jay was referencing. He's a associate professor at Fred Hutchinson Cancer Research Center. So, yeah, I'll check that out.
So anyway, I mean, he's harping on the complexity of the immune system. But anything that he's been trying to hit you on, he's not supported with any points. So
yeah, he kind of just started saying, "Well, we're not going to agree," and then getting upset, and
that you didn't agree.
Yeah, I'm not sure what he was expecting.
Yeah.
Yeah, I'll look through this talk. Yeah, I'll do it later.
Right. It's Clemens again makes a great point that the mRNA is degraded in days, and you'll see side effects from the antibodies within weeks.
Right.
As we've seen through decades of vaccine research, the side effects appear pretty rapidly in the short term.
Yeah. Laypersons like myself have some concerns about the unknown long-term effects of the vaccine. No, I'm not a conspiracy nut. No, you don't have to be a conspiracy nut to be concerned about that. That's a perfectly reasonable thing for someone to be concerned about. But as far as we know, throughout the history of testing vaccines and their safety and efficacy, almost every single adverse event caused by a vaccine emerges within 6 to 8 weeks of the dose being given. And that's because the adverse events are almost always caused by the immune response itself. And your active immune response to that encounter, it doesn't last. It doesn't last that very long. So once you're past that window, your immune system's not going to be. Your immune response to that antigen is not going to be causing many problems. So you know, we do these 2 to 3 month phase 3 studies to look at safety and efficacy, but then sometimes when we roll the vaccine out to millions of people, really, really rare events, like 1 in a million events, will only show up then. So if that's what you mean by long term, you know, that's also a valid concern, but over 6 billion doses of these vaccines have been given worldwide, and the time from now to the first vaccines being given has been over a year. So really, there's a ton of data to say they're safe.
And I love it when also he went back to there. It's like you were just like, well, we can't use FB, Facebook as a data point
That
because
was, oh man,
these
that
are
was
anecdotes. And then you're immediately like, well, what are you going to say next? That we got to go to VAERS to get this? And then he literally just said right after that, like, well, but then you're ignoring all the data in VAERS. And he's like, no, this is not data.
yeah,
No.
yeah. People,
Um, so what I understand about VAERS is that there's underreporting of vaccine side effects in VAERS, right? And at that level, statistically, if you put all of humanity in a grab bag, um, and then pulled out a random swath of it, you expect within a couple of weeks that some of those people are going to die. Statistically,
hmm,
it's just
yeah,
a sad reality of the fact that we have a finite life.
hmm,
And so at that rate, the amount of people who would be submitting to VAERS after they've died in a couple of weeks um, would match that rate, right? But there is actually not that many people in VAERS. So it's even lower than what you would expect by a random grabbing of the population,
hmm,
right? Because some of
right,
those deaths are obviously not caused by the virus, therefore it's not reported as such. So
right,
when they say VAERS is underreported, what they're meaning to say is that side effects are being ignored, but what actually it is no, the stats are suggesting that there should be a certain amount based on how many people would die in that predicted time period after getting a vaccine, and that amount's not showing up in VAERS because people aren't buying into that car accidents were caused by this person getting a vaccine.
right. Yeah, and another aspect to that is a lot of the underreporting that they talk about also has to do with super minor side effects. So, for example, like if someone gets a shot and they have a sore arm, that's an adverse effect.
Right.
But how many people go and report a sore arm to VAERS? I don't. No one really cares. It's a sore arm. So, but they count that as like, yeah, oh, it's underreported. Like if it's not super serious, people aren't going to report it.
Right.
So that's what they mean. Whereas really serious adverse events are most likely to be reported because if you go to the hospital, for example, for an adverse event to a vaccine, whether or not it's actually caused by the vaccine, if it was shortly after, that doctor is required to submit that to VAERS as
Right.
an event, and
Such
that's
as which,
yeah,
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 such as blood clots, which was just brought up in the chat.
right,
And
right,
if that caused an increased case, then you would see that
yeah, and
because those
we
are reported.
and we did for the J&J and AstraZeneca vaccines for a little bit.
Right.
I mean that they detected a 1 in a million event, so they monitor this stuff
Hmm.
really closely, and they really do follow up on those reports. In that case, it just happened to be a real, extremely rare event that is hundreds of thousands of times more likely to happen with a COVID infection.
And the
You're,
same thing with myocarditis.
Same thing, yeah.
Yep.
Myocarditis exists in the population. It happens for a multitude of reasons, and it's way more likely that you'll get that with a COVID infection as opposed to the vaccine. And if it happens with a
I mean, it should be
vaccine,
monitored. Yeah, it
what's
should
that?
be. It's monitored. It is monitored, just like the case with the J&J vaccine.
Hmm.
And there was a pause put on it so that um, the proper controls and follow-ups could be assessed, and then after that assessment was determined to be a negative,
Hmm.
then they move forward. You'll get spikes in the data.
Right. Uh, let's see. Okay. Pseudouridylation decreases mRNA immunogenicity. So it actually it still it stabilizes the mRNA by decreasing the overall immunogenicity, but it does still help recruitment of dendritic cells, which are going to be important for antigen presentation
Right.
once the spike protein is there. So that's kind of what I was trying to get at with it's a balancing act. You dampen some aspects of the innate immune response to the mRNA being there, but you still encourage parts of it that are enough to make it a good adjuvant. Are you going to vax your kids for COVID? Yes.
Absolutely.
Yeah.
I mean, I think I was talking to you once before about the seminar that Dr. Rhonda Patrick had, and I think she is sometimes absolutely spot on what she says. And she said specifically, you have a choice as to whether because this pandemic is becoming endemic, and at some point in the future, you're going to come across it. So do you want your immune system to be primed, or do you want to come across it without any help at all?
Right. Right.
It's not like you get a vaccine and the vaccine does the work for your immune system, right?
Mm-hmm.
It just allows a priming of your immune system to then go through and develop polyclonal antibodies and T cell response and all of that.
Right.
So,
Yeah, I always like the analogy that Rich Condit gives, which I recently heard him say he got it from somewhere else, but for me, I get it from him, where it's like if you get vaccinated, it's like having a fire extinguisher in your house.
mm-hmm.
Do you want, if you have a fire in your house, do you want to be able to put it out really quickly yourself, or do you want to have to wait and call the fire department for them to get there? You'd want to have the fire extinguisher there so that you can put it out quickly and not have
Right.
a ton of damage to your house.
Yeah.
And that's like what the vaccine does. It allows you to be ready for it when it happens.
Templar, are you saying that no kids have ever not suffered from having COVID? He's saying kids don't need the jab, and I think he's wrong.
What's it now?
But he just Templar, who's been in the chat for
Kids
a
don't
while.
need the jab. They kind of do. Kids suffer from COVID more rarely than adults do, but they do suffer from it. And the more COVID spreads throughout the young population, the more hospitalizations, the more deaths you'll have. And how many are okay with you? I mean, I know people whose kids have been in the hospital with COVID. It's not a good time. They were really traumatizing experiences for those people.
No, I can imagine that wouldn't be great.
Yeah, I mean, if my daughter was in the hospital with COVID, that would be terrifying. Like, why take that chance? Even if it's 1 in 1,000 kids who have a hard time with COVID.
Mm-hmm.
I mean, it's like if you put a bullet in a 1,000-chamber gun, spun it, and
then
put it to your kid's head, pulled the trigger, maybe they'll be fine. But this is an infectious disease, so you're not only doing it with them, you're doing it with the next person. Who does your kid pass COVID on to? They also have to pull the trigger and see what happens. And it's just a chain until someone gets hurt. So it's not worth it. It's best to vaccinate. And children absolutely do spread COVID. There was a paper, actually I was reading it yesterday, talking about how kids are reservoirs for SARS-CoV-2. Their viral RNA levels get just as high as adults do, and they did TCID 50. They actually measured infectious virus, and it's they actually saw that kids are spreading infectious virus. So I will. Do I have that on? Do I have that here? I do. I'll link it in the description.
Okay. Yeah, for me certainly. For my daughter, I would certainly get it. I wish she was in the age category that she could get it, because it makes me nervous. Analogies are disingenuous. No, it actually is pretty okay. Dice.
What
you
is
don't
it?
like the gun analogy? He says the gun analogy is disingenuous. It's pretty spot on, actually.
Yeah, I mean, it's pretty much because you're taking a chance on whether or not your kid is going to die. That's a possible outcome, even if it's
Or
rare.
be injured.
Even if it's rare, that's the chance you're taking. So why not, protect them? Why not lessen the chances?
Kids are spreading but not suffering. Again, no.
They are.
Determined evidence have shown that kids are suffering.
Yeah, they also.
Right, and then he's in the chat saying, like, long-term consequences about the vaccine, without a care in the world about unknown long-term consequences of COVID, which we already know.
Yeah.
Long haul COVID is certainly an issue.
Yeah, and you also can have MIS-C with COVID in children, multi-system inflammatory syndrome in children. That's been happening a decent amount. It's not just if you die, it's suffering these long-term illnesses that are going to be pretty traumatizing for your kid. What's more traumatizing for your kid, getting a vaccine, wearing a mask, or going to the hospital?
Right.
Or losing their caregiver, for that matter.
Yeah.
I'd say those things are more traumatizing for a kid than those other things. And no, I'm not experimenting on my child by giving them a vaccine. The vaccines have been tested very extensively. These COVID vaccines are arguably now the most studied vaccines in the world.
Yeah, I mean, the amount that was given in such a short period of time, and then the follow-up since then.
Yeah, I mean,
where's it been a likely mechanism that these vaccines have caused injury, and it hasn't really materialized.
Right. Yeah, and this person saying that it's possible for children to die from the vaccine, both are possible and rare, but one is much less rare than the other.
Yeah.
There's actually, I mean, you'd be hard pressed to find a confirmed death in a kid from COVID vaccine. You'll see stories, correlations, but nothing confirmed.
Right.
And it needs to be confirmed if you really want to say that. But you can find plenty of examples of children dying. MIS-C is caused by the spike protein. Well, it's not entirely clear what causes MIS-C in children. Last I read about it, there were ideas that leaky gut caused by the COVID infection leads to this MIS-C event later down the road. But as far as I know, it's not completely understood what actually causes MIS-C in children.
And it's certainly not an unconformationally active version of the spike protein that's in the vaccine. And again, it just circles back around to you're completely okay with your child coming into contact with conformationally active spike protein from the virus in much higher quantities. In a much less controlled situation.
Exactly, yeah. An infection is totally uncontrolled. All right, people are starting to talk a lot about kids and COVID vaccines, but I mean, yeah, this stream has been going on for a while now.
Sure. Yeah, I think you've basically answered and re-answered everything that he was trying to get at in the Gish Gallop manner that he was trying to address it in. So
Yeah, and I'm kind of clicking through this Trevor Bedford
Yeah.
talk. I can't. I'm not listening to what he's saying, but I'm kind of just looking at his slides. Um, just just reading from his conclusion slides at the end. One of his points says rates of adapted sequence evolution in SARS-CoV-2 have been about 5 times that of H3N2 flu. Expect this to slow down, but suggests this virus will be generally quite adaptable.
Mm-hmm.
So I mean
that's completely in line with everything that I've seen in the database. So,
yeah, I'm not really seeing any slides that explicitly say this is because of the vaccine. I think he's just saying that the spike protein is evolving quickly, and which it's going to do no matter if we vaccinate or not, because people are going to get infected, they're going to have natural immunity. The virus is always going to be fighting the immune system. That's a selection pressure. So either we slow it down or we just kind of lay down and take it. So I suspect that he's just misinterpreting Trevor's work. I'll send Trevor an email, see if he responds, see what he says.
Okay.
But
S1 is evolving, but not S2.
S involved. Okay.
It was just a chat comment. So
the S1 has the RBD.
Yeah.
So I mean that makes sense that it would be evolving faster. But again, you develop antibodies and T cell immunity against the whole protein. So
I would expect that.
Yeah.
That Trevor doesn't reach that conclusion as pointed out in the chat. So.
Okay.
Yeah.
Yeah. All right. Well, this was an interesting. My first rage quit. That was fun. Yeah, that was
usually rage quit if you're ahead.
Yeah.
By very far.
Yeah, that was fun. Well, interesting talk. Thanks everybody for staying around and chatting. I know I didn't do that last time. I kind of was it on. I think it was on a Saturday. I think I was very tired. I just wanted to get to bed.
It was a long one.
Yeah, I usually have
the
to.
last one.
Yeah, I usually have to wake up early with the kid, and that discussion was. I wasn't too excited about it. It was pretty lame, in my opinion. But this was fun. Thanks for coming on, talking at the end. Appreciate it.
I think like you needed somebody to kind of reflect off of the dumpster fire that happened on his side. So
yeah.
Yeah, hopefully he gets on and just like calms down a little bit and bring some data, dude. Just bring the data and let's take a look at it. You know.
Yeah, I mean, I had a list of links ready to go, and I wish he had done the same.
Yep. So okay, well, I'm signing off.
All right.
Good job, Dr. Wilson, as always. Best of luck.
All right, I'll see you later, man. One more question: did James Corbett ever respond to your videos? No, I haven't thought about James Corbett in a while. He'll probably make another appearance on my channel eventually. All right. Well, thanks everyone. This was fun. Hope you all enjoyed it. Hope it was informative. I'll link the relevant studies in the description. If I don't do it tonight, I'll do it tomorrow. But thanks for watching. See you guys next time.